It takes a Oh, we are live on YouTube, I think. And we also need to close the door. Oh, it has a little delay. Whatever. Enjoy your protein bar for the 12K this week.
Thanks.
You were running? Or you're running later?
Morning Friday.
Cool.
Yeah, we're gonna wait a little bit. Do you know whether I will get or the people who join later whether they can still check the chat and link? Will they?
I think not.
Well, you can already take a look. Do you mind if I make you co-host so that if people come in, you can just admit them? Because
Yeah, go for it.
hey, Dordo. Because I will present
Hey.
at the same time. A slightly stressful situation given that I prepared the paper today in the morning.
Who's presenting today?
Me.
Oh, cool. Stephanie Forkel.
Yeah. Yeah, it was slightly interesting, stressful situation yesterday. Because Steph was like, "Yeah, Anna, you ready for tomorrow?" I'm like, "Yeah, all good." And she's like, "Yeah, who's presenting tomorrow?" I'm like, "I don't know." And I was like at the airport because my boyfriend's flight got canceled. So we were like, "Oh shit, what's supposed to do?" Like it's all like a little bit chaotic, right? And then I check at the timetable and it's me presenting. I'm like, "Oh my, oh no."
Been there.
Yeah. So so don't be too harsh on me today.
Yeah, would be harsh on the paper.
And let me know also when I'm like at 50, like at 20 minutes, like 9:50 or something, if I don't have that. And yeah. Actually, you know what? I can set the timer. That will also work. But that's like it's not as cool as, ending the presentation on time without the timer, having your internal clock. Eduardo, I'm so happy you're joining today because you're gonna retrieve your knowledge from what you learned from Oliver Turnbull in Bangor. I need you. I'm counting on you.
Yeah, it depends. Is it about the brainstem or the cranial
That
nerves?
is absolutely right.
Oh no, I think
It's
I
the
already lost
brainstem.
those. The
No, it's good.
brainstem.
You just need to
Oh
know.
no.
You just need to know three structures, which you know.
Depends on the resolution we are speaking of.
Are we only four people? We're gonna wait one more minute and then we're gonna start.
Yeah, I think Lilith has a course at the university, so
Right.
probably not. Steph, who knows?
Who knows what she's doing?
So
Yeah, cool. Muga is coming. Who knows what Steph is doing? Steph is probably still in London. Is she in London? No. Yeah, yeah, she is. She is. She says it's too early, and UK is always behind. That's how I always know what the time. So that is like 1 hour behind our time. Because we're here used to say back in the days, yeah, UK is always behind. Dipping into some political and social issues probably as well.
well, if you ask them, they are exactly on the 0 time zone, so.
Right, yeah. But that's true. But that's because the time zones, like the dude who invented them, he just said, "Oh, it's gonna be in my little town, the 0.0."
I know.
I probably would have done the same thing, to be very fair. But like naming the brain structures after yourself. It's the
well, we have the Rolandic fissure, the Sylvian fissure. People did that.
exactly.
The frontal slant. The frontal slant.
Where's the joke?
It's not a person, but that's okay.
It's a dude, apparently.
Really?
I didn't know either. No? I'm wondering where we're gonna have the Thibaut de shot in 1, 2, 3, like the SLFs.
I gave the name frontal slant to the frontal slant.
Oh, really?
That was why it's a joke.
Why did you call it like frontal Michelle or something?
I'm not like that. Like that.
That's a good name though.
Right. Okay. I think we're gonna start. Does everyone have the link in the chat? Can everybody see it? Okay, then I'm sending it again. So if I was right about nobody can see it who joins later. All right. So please mind my exhaustive highlighting of this paper. It's like those memes, just highlight the most important thing and then like the entire page gets highlighted. So today I'm gonna talk about the brainstem, as it had already guessed correctly, namely about an article called Integrating Brainstem and Cortical Functional Architectures by Justine Hansen as a leading author, who is a rising star in the field from McGill. And so she and her colleagues, they took a closer look at the brainstem that is like typically excluded from in vivo human brain mapping, and therefore we don't have that much information on it. And what they did were that they used a 7 Tesla fMRI and derived basically a bunch of information on the functional connectome and also on the functional connectome, they looked at 58 brainstem nuclei, stems basically covering the entire brainstem, spanning from the midbrain, the pons, and then medulla. And what they got out, what we're going to talk about, or what we're going to dive into a little bit more, is that they identified a set of integrative hubs, basically, in the brainstem that have a widespread connectivity to the cerebral cortex. And we have already like identified hubs around the cortex, but not really for the brainstem, because again, it has been like not really looked into. Due to several difficulties studying the brainstem, starting with like the nuclei being very small and also the depth basically of the tissue or of the structure, not allowing for a very very high signals. But we're going to talk about that a little bit later. So they did not only identify those hubs of like those integrative hubs of functions, but they also looked at like patterns of connectivity between the brainstem and the cerebral cortex, and they checked the neuropsychological oscillatory rhythms to basically compare them to the known neuropsychological oscillatory rhythms, and looked at like the cognitive functional specialization at the unimodal, transmodal functional hierarchy. And at the end, they also like validated this entire thing that they did in the 3 Tesla data from the same participants. So they start their introduction with like explaining again why it's why the brainstem has been not ignored necessarily, but again the signal quality in the cortex is highest, so you don't reach the brainstem as easily, but it's still very crucial, as we probably learned very early in our career stages, for like survival, since it's like one of the oldest evolutionary oldest structures. It's very crucial for survival and like consciousness, and it integrates basically a bunch of signals across the nervous system. So to just give you a little not background, but like a little starting information. So we have the brainstem nuclei that they looked into, and they had like they identified or they studied 58 brainstem nuclei. You can see them basically here in the entire brain, here the parcellations. Right, so first of all, they used this whole brainstem atlas to identify those 58 nuclei, to then check their connectivity of the specific nuclei with the entire cortex. And to just, looking at the brainstem itself, you can look at the functional, they check the functional connectivity between the brainstem and brainstem itself, like within brainstem, the brainstem to cortex connectivity, and also looked at the within cortex functional connectivity. And as you can see here in the figure D, so this is on the y-axis you see the density, on the x-axis you see the functional connectivity, and what becomes evident here is that the brainstem to cortex functional connectivity is higher, basically, that you can see here, than within the brainstem. So one of the key things that they identified here is the functional connectivity to the cortex itself is higher. And if we look at the distance, so what we can see from the figure E here, it's basically a scatter plot of the functional connectivity between regions as function of like Euclidean distances. So we'll see the Euclidean distance and the functional connectivity. And when you compare the brainstem to cortex connectivity, it tends to, like, even with increasing distances, it does not lose of functional connectivity as the cortex itself, right? So this is also like one of the key things they identified here. So to dive into the main results that they found out, so first, as we just talked about the brainstem cortex functional connectivity, that was basically identified, and then they took a more detailed look at which nuclei of those 58 nuclei that they studied, which nuclei basically had had what kind of degree of connectivity to the cortex. And we can see that in the figure A. So based on the size of so this is basically, you probably already see the brainstem structure, and all these names are the specific nuclei of the brainstem, and the weighted degree is basically the strength of the connectivity to the cortex, and the size of, like, the darkness of the circles, and the size of the circles is indicative of the strength of the connectivity. And so you can identify already, like, which structures of the brainstem or have, like, which main nuclei that are connected to the cortex itself. So now, Eduardo, it's your turn. Can you name me, like, just looking anybody else as well, right? Very welcome. It's just something we learned recently at Visceral Mind in a summer school. So you can see the structure of the brainstem, and can you name me, like, one of these like, look at all the nuclei, and which one is, like, the main one, for example, for the midbrain, or which ones are the ones that are the midbrain nuclei that are connecting basically the substructure of the brainstem, the midbrain, to the cortex.
But there is a bunch of them. Already in the midbrain, I can see the the reticular formation, the
Awesome.
periaqueductal gray. Yeah,
Very good, very good.
way too much.
Okay, do you want to continue? What about the pons? What is, like, the main nucleus of the pons that is connecting the brainstem to the cortex?
My anatomy knowledge is starting to fail as we leave the brain. But I don't know. It's it's really the point where you start to meet all the adrenergic nuclei, but and they have, like, vast connections through the cerebral cortex. But yeah.
You just so basically what we need to know or follow here is that this is basically a part of the midbrain, right? Here we have the pons, so the main nucleus in the pons is like the pontine and caudalis that is connecting to the cortex, and then we have the medulla here underneath. So basically, these are the three structures that I was looking for. But thank you very much. So right, so that's what they did on the nuclei level, and also one thing, I don't know if we'll have time to look into that, but it's like if you look into the paper, really interesting thing. So locus coeruleus, which is known as like the integrative nucleus of the brain. I don't know if I'm exaggerating or not, but you can see that like locus coeruleus is not. It's pretty small, no. So keep that in mind. Keep that in mind, and
What she said.
keep that in mind for eventually the discussion. Okay, I see that I don't have too much time left, so I will proceed. They looked at like gradients, and also, so maybe this is maybe also the C and D figure. They're interesting to look at. So here in the C, we see the classes of laminar differentiation, and what did I want to say about that? That it's like you can see that they're like pretty exclusive in that sense. Hold on, I will segregate. Oh yeah, so the one that are most functionally connected with the cortex are spatially segregated in line with the theory that hub placement optimizes the trade-off between distance and efficient information transfer. And that's basically what you can see here in the C. And yeah, interestingly as well, like the site architectonic classes, like the insula and the limbic class, they have the highest connectivity, cortex to brainstem connectivity, and then we have like the primary sensory motor areas that have like a lower one. Okay, another like one of the last I think, okay, not the last thing I want to talk about, but one of the main points of this paper is as well, so they looked at brainstem communities that are underlying the cortical function, what you can see here, and which is super cool because they used also like neurosynth decoding and looked basically at and identified those hubs of different nuclei, which you can see here, right? So they identified those brainstem communities that are associated with different higher and lower cognitive functions. So they regressed basically how they proceeded. You can see that in the figure A. I need to take a look though at the notes. So they regressed basically the brainstem nuclei connectivity out of this calculation because they wanted to take a look at like more fine-grained grain structures, and that's how they obtained, like, pretty much it's a very very easy explanation, but that's how they obtained those brainstem communities, looking or basically identifying different nuclei that are associated with different functions. So they correlated, I believe, the cortical weighted degree of the brainstem communities, and you have like a here, the maps that are color coded as well. So you can see here is basically the green one, and the most interesting ones, as like for me at least, were the green and the blue ones, which are the higher cognitive structures, higher cognitive, not structures, but abilities. And career patterns, right? So that's they looked at the cortical weighted degree patterns that they calculated here in C that you can see, and they correlated those weighted degree patterns with the information from Neurosynth, right? And that's how they obtained those correlations basically with different functions. And these are only the top 10% of them. Like in the supplementary material, they have more. But what's again the most interesting part is are the green and the blue Neurosynth keywords, because we have the higher order cognitive functions, and you can, if you look at back at the brainstem communities that they were derived from, you can see that the higher, so for the higher cognitive functions are, for example, identified here with the midbrain and also the pons, right? And the sensory functions, like more sensory functions, can also be found in the midbrain, whereas the motor functions, like movement, motor control, multisensory, and also perception so lower order cognitive functions can be found like more deeper in the medulla area. So this is something that's pretty cool also with how they visualized it. But there is so much more basically what they mention in the paper and what they discuss. But because of time constraints, I'm just gonna move on, because they also have this really interesting part of the work where they mapped the chemical architecture to those brainstem communities that we just identified, right? We have so just to, maybe we can name the brainstem community. So we have, they didn't give them names, but yeah, green and blue were higher order cognitive functions, and everything else was basically slightly lower order cognitive function and sensory related community, which is the gray one, sensory related. So what they did here, given basically that the cortex receives a bunch of input from like multiple neuromodulatory brainstem nuclei, they looked basically they try to identify the relationship basically between neurotransmitters and neurotransmitter receptors. So they checked the distribution of 18 neurotransmitter receptors and transporters, and try to look into how they basically try to map them to those communities, to those like brainstem communities. And what they found out here, so this you can see here, for example, these are all those receptors and transporters, and you can see the net, which is the most dominant receptor that is basically spreads across all the identified hubs, the communities, and it basically includes, as they say, the primary nucleus for norepinephrine synthesis, the locus coeruleus. Although, as you remember from earlier, from the figure before, from one of the first figures, the connectivity of the locus coeruleus was really tiny, as you remember from the little dots basically. And in the discussion they discussed that finding, which makes at the end it makes a lot of sense, because apparently locus coeruleus is only in like certain circumstances it becomes as important or as integrating as we know it to be. Okay, given the time, I just gonna wrap up and tell you so they're finding from looking at the chemo architecture is that cortical regions with similar scores along the gradients that we saw earlier are similarly connected within the brainstem, and the greater sorry, that was already the next thing. That's already the unimodal and transmodal cortex. But sorry, I will have to take a more detailed look. So I won't have the time to get into the last part, but yeah, this is basically what they found out for mapping the chemo architecture, and it's apparently it made it made a lot of sense because it mirrored the chemo architecture of the cortex as well. So, yeah, proving or showing that we have a very similar chemo architecture in the brainstem and the functional connectivity of the brainstem with the cortex. So it basically aligned pretty well. Yeah, I won't have the time to go into the unimodal and the transmodal cortical regions, but essentially what they found looking at those was that like a lot of nuclei were transmodal, and yeah, and it basically like all the findings remain consistent with when they extended the analysis to subcortical regions, and they also looked at subcortical regions, and they tried to identify hubs in subcortical regions, and one of the like very, very prominent hubs was the thalamus, which makes also a lot of sense given that the thalamus is basically like all the connections from the brainstem go through the thalamus in order to connect to the cortex. So thalamus must be a hub. Michelle is looking skeptical, I see that. So I'm sorry we won't have the time to look at the discussion, but have a read. It's super interesting, especially the locus coeruleus stuff. And I'm really sorry that I didn't have the time and probably also the knowledge to to completely summarize the entire article, but it's worth a read. And I see already the first hands rising. So Eduardo, once you start the discussion or a question, happy to take them all.
Yeah, thank you so much. Really brave of you of going down the rabbit hole of the brainstem. So my question, also Stefan is watching us, so we have to behave.
Ah,
My first question is about the figure 3, I guess, the one with the correlation with all the function, like the Neuroccino decoding.
there you go. Yeah.
Which it's really cool, but I wonder about what's the meaning, because correlating the brainstem functional connectivity to these maps of the like basically functional stuff in the cortex, and there is also some heterogeneity in the classes, in the communities they found out. And so I was wondering whether it's more a sort of epiphenomenon of the brainstem being connected to, basically it's like a bottleneck for the brain anyway. So what's the meaning of being uh, correlated with all these cognitive functions? Yeah, I don't know if it's a
Well,
question.
so firstly, it's the brainstem was not considered really a long time to be relevant for like higher order cognitive functions. And it's cool that you can basically, by just looking at the functional connectivity from the brainstem to the cortex, just like not mimic, but you can see that they are that you have some higher order cognitive functions as well in there, and you can identify hubs. So you see like different nuclei that are actually associated with higher cognitive functions. That is pretty cool. That's pretty new as well. And also like the functional connectivity itself of the brainstem is just not studied, or has been not really studied yet before.
Yeah, but so do they think that um the brainstem is involved in these functions, or that is more like a modulator on these functions, because this is the role we know for at least some of the nuclei in the brainstem, right? They have like a spread connections over the cortex, like I don't know, the adrenergic system or the serotonergic system. So I don't know what's their position. Do they think it's part of the processing or
Yeah.
it's yeah?
Yeah,
Okay.
so that's what they say also in the discussion. That's like one of the main or one of the intriguing things to which we would have I would have loved to have the time to discuss this. For example, the implication for neurological disorders, right? And the incomplete view that we have been that we had so far, or that we are being led by. So basically, integrating the brainstem, given also that the um neuromodul like chemoarchitectural stuff, and like the neurotransmitters and modulators, they are coming from the brainstem, as if I'm like not completely Michelle needs to probably say yes or no to whether that's correct or not, but as far as I know.
I was speaking with Steph, sorry.
Oh, yeah, so I absolutely, they think that is that's a part that is not has been that hasn't been looked into so far yet, and it's and yeah, part
Okay.
of the whole.
Cool. I think I'll leave like space for Michelle and
Yeah.
Muga also to ask questions.
Go, Michelle. I'm gonna mute myself real quick.
So I was thank you so much for the presentation. That's a very rich paper with a lot of analysis. And so I was speaking with Steph who's watching us via text message, and she was surprised that there was not so much contribution in language from those nuclei. And then I did a little bit of more research because my anatomy, the brainstem is not from yesterday. But I couldn't find the hypoglossal and the ambiguous nuclei inside the list, which will be the cranial nerve 12, 9, and 10. And so I was wondering whether some of the nuclei were missing in the original models that had been downloaded to do the correlation. And that's why there is an underrepresentation of language.
I am not completely sure, but they have this 1 second. Here, brainstem nuclear segmentation. There it is. So there is this probabilistic atlas of in vivo brainstem, which has apparently 58 nuclei. This is how they, yeah, these are the nuclei that they took. And they identified them by like looking at diffusion and T2 weighted images, also like double checked with neurosurgeons. So I'm not sure because I'm not familiar with the resource. So I don't know whether some of them are missing or not. Are they more brain nuclei than 58? Maybe these are the most, well, maybe these were the ones that are the easiest to segment. And yeah, so they have like those different like semi-automatic and like manual segmentations. I don't know if there are more nuclei actually. If someone knows how many brain nuclei there, I can Google that.
I think
Mm.
nobody knows. Like
Brainstem nuclei.
but Michelle, why would you think the cranial nerves nuclei would be related to language, for example, the production aspects?
It's because sorry, can you
Sorry,
just
yeah. I need to turn up the volume.
well so you see there's a card. I can hear myself, it's horrible. You see the cortical spine tract is connecting the motor cortex, but it's not connecting the motor areas are related to the face and the mouth that are going and stopping in the brainstem through a different tracks at the cortical spinal tract. So when people are showing the cortical spinal tract that is fanning onto the motor cortex, it is not supposed to fan up to the most lower part that would be the face and the mouth. And those, there is a direct connection then to nuclei in the brainstem. So I don't remember the name of the nuclei in the brainstem, but that's one of those that I mentioned just before. And that's why we know it's related to language. Now, as to how does it interact with those regions for language, we don't really know. That answer your question?
Yeah, thank you. So what tract would it be from?
It's a cortical pontine.
Cortical
fibers.
pontine, yeah.
Yeah. Cortical bulbar fiber, sorry. Actually.
So there is this where they took the nuclei from. It's this NITRC, Neuroimaging Tools and Research Laboratory. There is this thing called Brainstem Navigator. I am sending a link in the Zoom chat if I can find the Zoom chat. So you can take a look potentially here. I'll send it to YouTube as well. And yeah, happy to take Muga's question because we need to wrap up.
Yeah, okay. Thanks, Anna. This is very interesting. I'm definitely going to read. I just wanted to clarify something. So they are looking at functional connectivity using fMRI, right?
Yes, that's right.
But they also mentioned like the oscillatory dynamics. So what is that about?
So this was the oscillatory dynamics they took from the PT, I think. So reflex conjugate ontologies. This was, yeah, 1 second. I'm gonna do the trick here. Oscillatory rhythms.
Yeah. So I thought like some EG or something is
MEG.
MEG, yeah.
MEG, yes. So right here it is. Therefore, it is that the whereas gradient is aligned with the measurements of the cortical dynamics, which are the neural oscillatory rhythms, and for that, yeah, they correlated MEG distributions for frequency bands.
Okay. So no fMRI like connectivity analysis.
Well, that was before, right? So they looked before at the fMRI connectivity analysis, but for the oscillatory rhythms, they needed to
Also,
take
they do too. They do both. Okay.
Yeah. They
Okay.
do like a bunch of stuff. So they do
Yeah. Okay.
fMRI, the MEG stuff, then for the for the chemo architecture of the brainstem communities, they collected chemo architectural data. I'm not sure how that actually works.
Yeah.
But yes, so they do a bunch of stuff.
So I just like probably they choose the nuclei as a like for the fMRI connectivity as a seed region and look at the connectivity of that seed region to all the different parts of the cortex, probably, right?
Yes.
But when they talk about like unimodal and transmodal, they first define the unimodal and transmodal regions on the cortex, and then check like which nuclei have more connections to that, specific unimodal or transmodal region in the cortex.
So they have the as we can see here. Sorry, hold on here. So they create those maps of the weighted degrees of the brainstem communities, right? So they create first basically specific maps where, as you rightfully say, I think they take each nuclei and its whole connectivity to the cortex, and then they use neurosynth. So they basically correlate the pattern that comes out of there with everything on with neurosynth, and I think I don't know how many 123 cognitive and behavioral activation maps of neurosynth. The neurosynth is fMRI, right? So they basically they do correlate fMRI with fMRI.
And the conclusion is that most of these nuclei communities are basically have mostly like transmodal connections. Is that the
So this is the part that
the
we
take home?
didn't have the time to discuss properly.
Mhm. Okay.
It's here somewhere. So, right, so the question that they want to answer is, right, that which brainstem nuclei are more functionally connected with unimodal and transmodal regions. And for that, they calculate what we just talked about, the weighted degree of the functional connectivity from both negatively and positively scored cortical regions in the brainstem. And what they find is, yes, the unimodal brain regions are most connected with, with certain, like with caudal brainstem nuclei in the medulla, and other
High function.
transmodal regions exist in the midbrain and the midbrain and the pons. And then if we go back to the hubs that we can see, we discussed here, right? You can see the hubs of the midbrain, the hubs of the pons and the and the medulla. You can basically trace it back to the nuclei, to the specific nuclei
Okay.
that are involved. Okay.
Okay, thank you.
Sure, we have one more question from Mikolai Pavlak on YouTube. Can you comment on functional connectivity in bold of the nuclei that have high iron content? For example, red nucleus is going to have very different signal because of high susceptibility. I don't know. I'm sorry. Sorry to not being able to comment on that. I don't know how the iron content is influencing the fMRI signal in bold. Michelle, maybe you know?
What was the question?
Functional connectivity in bold of the nuclei that have high iron content. For example, red nucleus is going to have very different signal because of high susceptibility. I don't know that much about how iron content is related to
Well,
the signal.
If you change the baseline,
You're muted, sorry.
If you change the baseline of the level of the bold signal, which I'm not sure is sensitive to iron, but because I'm not a physician of MRI, that's a question from Tomato, yes. But if you change the baseline, it will not change the correlation, which are independent of the magnitude of the signal, because that's how correlation works. So it should not be influenced.
Okay, cool. Thank you very much. I think that if there is no other question, then I will stop sharing my screen and leave you with that little teaser of a paper. Again, apologies for not being able to go through it completely and to just leave you with some bites of information. But I still hope that it was somewhat informative, and thank you for joining. And on that note, have an exciting week in science and take care.